Okay, I think we can get started. So thank you everyone so much for coming today. We're very excited to have you. My name is Sarah Shepard. I'm from the career team at Mercy College. And we are really happy to host several representatives from Regeneron, and they're here to speak with you. The agenda for the day is on the screen, and we're going to start today with an introduction video about Regeneron, and then we'll have our three panels. And at the end of the three panels, we'll have time for Q&A with everyone. So I'm just going to quickly stop sharing and get that video up.
One of the things that's made Regeneron really unique is its willingness to invest in cutting-edge technologies.
We're one of the few companies in the world that go all the way from foundational discovery
to research, from research to development,
to manufacturing and commercialization. We do that all at our campuses in New York, New Jersey, Ireland, and the UK.
It's really cool because we go from the really big thought, the really big idea, all the way to seeing the impact on a person.
One of the things that distinguishes Regeneron is that we are a founder-led organization.
Both Len and George are scientists, and they've really set the tone of focusing on science from the very beginning.
And it spreads through the organization in terms of our mission, our vision, our activities, our behaviors.
We're one of the unique places where we've put such a big emphasis on human genetics and deep biology. Our genetics research is focused on looking at differences in our genes and connecting that to diseases.
We have a capability of combining human gene sequence data with electronic medical records.
We have a rich genomic database, and we apply that across our organization, looking for new insights and to advance our research programs.
We've invested in a set of core technologies we call the VelociSuite technologies. These include things like VelociGene, VelociMune, and together this suite of technologies gives us an engine for drug discovery. We can use that engine again and again and again to produce new drugs. Our VelociGene technologies give us an unprecedented ability to precisely manipulate the mouse genome and work very rapidly. Likewise, the VelociMune technology gives us an unprecedented speed in producing new drug candidates.
So at a high level in drug development, there are really three key questions that we need to answer, right? The one is, does it work? Is it safe? And the third is, can we produce it? Before we can even start to explore whether it works, part of the role in preclinical development is to establish whether it's safe and also develop a manufacturing process by which we can produce it. So I see ourselves as a link between the discovery world and the manufacturing facility where they will produce the drugs that we can test in clinical trials and ultimately also do commercial production.
Once we've completed preclinical research to confirm safety, we work internally with our medical experts to develop our trial designs. We take those designs and work with healthcare providers across the globe to help us implement clinical trials. After decades of research at a lab bench and years of clinical trial work, we take tremendous pride in being able to determine whether we've made a difference in patient lives. After completion of our clinical trials and data analysis, we then take that data to regulatory authorities across the globe so that they can give us our stamp of approval. With this approval, we work with our manufacturing group to switch into high gear.
All of our drugs, whether they're for a clinical trial or a commercial market, are made across our manufacturing footprint in both Rensselaer, New York, and Regeneron, Ireland. Our quality standard is very simple. We want to make something that at the end of the day we would put in ourselves, our children, our siblings, our parents, our grandparents. And we have a just tremendous responsibility to get things that are safe and efficacious to those people.
commercial organization at Regeneron is responsible and accountable for the really important step of making sure that our medicines get to the patients who need them. We want to lead the industry in competitive commercialization, and that activity is powered by our science.
It's really a dream to work at Regeneron, where number one is patients and using science to help them.
I love the commitment to science. I love that we are foundationally a deeply ethical company.
At Regeneron, we study genetics.
We build cutting-edge technologies.
We develop biologic medicines.
We conduct thorough research.
We follow the science.
We bring innovative medicines to patients
Because the potential
To use science
To improve human health is endless.
And we're driven
by what we've yet to discover.
Okay, awesome video. I hope you all enjoyed that as well. So we are going to move on to our very first panel. Jorlene will be moderating that, and this is with scientists characterizing future therapeutic drugs. So Jorlene, take it away.
Thank you, Sarah. Hi, my name is Dorleen. I am an Associate Director of Career and Professional Development at Mercy College, and I'll be moderating for this first part of our discussion with Panel 1, Regeneron Scientists Characterizing Future Therapeutic Drugs. I'd like to ask each panelist to briefly introduce yourselves to us.
We start with Irena.
Yes.
Hi, everyone. My name is Irena Lovrick. I've been working at Regeneron for about almost 10 years. And what I love about science is that I feel like I'm always learning something new every single day. And that's always exciting.
Kristen. Oh. Kristen, you're on mute.
Sorry about that. Hi, I'm Kristen Libertini. I've been working at Regeneron for about 10 years. What I really love about working at Regeneron is that we work with cutting edge science, and every day is an adventure, and that we really get to see that we're helping people, and that's something that you don't always get to see. And like with coronavirus, we're helping people every day, and also with Ebola, we really get to see that we're actually helping people. So that's something that I really love.
Willy.
Hi there. My name is Willy Ramos, and I've been working in the company. This is my second time at the company. I've been here a total of maybe like 1 year and a half. One of the reasons I love working here is because I still get to do research in a pharmaceutical setting, and that's a very, very it's not happening across the entire industry, as you will probably learn in the future. There's very few places that actually do basic research and discovery research, and we are actually one of the lucky ones who get to go to a lab and discover new things and have the tools to perform them. And so I feel really grateful to be a scientist in this place.
Sangrim.
Hi, my name is Sangrim Bosley. I've been with Regeneron now for 5 years. What I enjoy about Regeneron is like what Kristen has mentioned, that it does tackle like really important issues like COVID, Ebola, and as well as gives us an opportunity to work on science in a fun way with the technology-based assays that we've been incorporating, and the collaborations amongst different groups within the company, and learning from each other and expanding our knowledge base from what we already know in our own field, but in different fields as well.
Diana.
Hi everybody, I'm Diana. I've been at Regeneron for 21 years, long time. What I like about science as a whole is, and at Regeneron especially, is it allows me to be part of something bigger than myself, right? Obviously, we are helping individuals, we are helping patients, improving quality of life for not just everybody, but our loved ones, et cetera, et cetera. So it's definitely a very rewarding kind of experience, that's for sure. Thank you.
Carla.
Hi, I'm Carla Springer. I've been at Regeneron for almost 5 years now, and I really love that our company is, like you've heard, cutting edge technology. So we're doing new exciting science every day. There's never a boring day at Regeneron. We're always at the cutting edge of technology, developing new drugs for impending diseases, and it makes for a really exciting career. I really love our company for that.
Ishita?
I am Ishita Chatterjee. I have been in Regeneron for 10 plus years. It'll be 11 years next year. And like Willy just said, this is my second time in Regeneron. So you can imagine I came back because I like the people and the science. And that's, I would say, the driving force for me to stay in this company is the people I work with are great, and the science we do is cutting edge and also very rewarding, very satisfying, and it's like it's a wonderful place to work. And in between, I have worked in other companies, but that's why I'm saying that I can say for sure that this is one of the best places to work for. Thank you.
Thank you so much for sharing that with us. I'm going to go ahead and start with some questions. Irena, can you talk to us about your career path from college to where you are now?
Absolutely. So I attended Northeastern University in Boston, and I had started in 2005. And during school, I decided I wanted to try, I already knew I wanted to go into science, but I wanted to try one of the campus labs. So I went my second year. It took a year to just kind of adjust, and then, or sorry, 1 semester to adjust. And then the second semester, I joined a lab, and it was very interesting. It was a circadian rhythms lab. And I learned a lot of different, very academic lab techniques, I would say. And it was very interesting. And so I stayed throughout my whole undergrad to in that lab, which was very good. So, while I wasn't going to classes, I would just hop into the lab, see if I needed to help out with anything. And that was really fun for me, just being in the lab. I just felt really, really happy to do it. Northeastern University also has something called a co-op. So it's usually a 5-year university, but I was able to still finish within 4 years and 1 semester. So they built in internships into their curriculum, which is, I thought it was wonderful. But I know a lot of universities and colleges offer internships, and I think that honestly was one of the best decisions I made, was to participate in the co-op program. So during co-op, I actually went to a pharmaceutical company, which is a small biotech company in Watertown, Massachusetts. And there I learned everything about behavior research. So I did a lot of the behavior assays, on mice. So I found that very interesting, and I really liked it, so much so that I asked if I could stay on for the weekends. So I would have like a 2-hour commute, and I would go 2 hours one way over the weekend, perform anything they needed me to do, and then 2 hours to come back home. So I really did enjoy that. In addition, I also I wasn't sure I wanted to be pre-med. And so I started volunteering at Children's Hospital during my 3rd year at Northeastern. And that was also very fulfilling. And I was able to do it all the way until I moved to New York. So that was great. All that experience really added up for me. And so when I graduated in 2009, it was right after the big market crash in 2008. So finding a job was very difficult. And I'm sure a lot of people are thinking about a similar situation now. My internship was the thing that actually helped me out, because my old supervisor reached out to me when they had someone leaving for maternity leave and asked if I wanted to fill in. And that's really how I got my foot in the door to eventually become a permanent employee for In Vivo Pharmaceuticals, which is no longer there, unfortunately. So after working there for about almost 2 years as full-time, it was such a small biotech company. It was great because I was able to learn everything from the in vivo all the way through to the in vitro assays. So I really got to see all the different parts of, basically developing and exploring a new therapeutic. We were doing small molecules, and I was more of a neuro company, so they were focusing on neuro diseases. But finally, I decided it was time to go, and I wanted to move back to New York. This is where I'm originally from, and which is where I grew up. Sorry, and then I lucked out and got a phone call from Pam to see if I could potentially join her group in the pharmacokinetics department, and it was a great break because it was nice to start in a new company, and I found big differences between the biotech, and the more of a better pharma, like a bigger pharma. And that was really nice to see that, they had so much. We at Regeneron, we have so much support. We have so much more ability to not worry about, oh, do we have the right things and tools in place? Really, it's more focus on the science. We'll take care of everything else. So that was really nice. But that's it. And I've been at Regeneron for, like I said, almost 10 years.
Wow. Thank you for sharing your career path with us. I'll open the next question up to the entire panel. What would you tell yourself back in college, knowing what you know now, especially about the career path that you've chosen?
We just shout it out.
Yes, go for it, Diana.
Pay more attention in chemistry. That's what I would tell myself, quite honestly. What I would probably tell myself is maybe, and the landscape for me 20 years ago was a lot different than the way it is now for students, but I would probably tell myself to maybe think about internships a little bit more. I see new employees coming in that have those internships or those extra set of skills, a little bit more experience than some of the other candidates. And I think they do a little bit better, or they're given not more of an opportunity, but having that little extra experience. So if I were to tell myself, besides paying attention in chemistry, is to look for those internships. I think they do help out tremendously. Again, like Irena said, right? Her internship helped out pretty well. So look for those internships. They are out there.
Can I add something? And just to follow up on Diana's comment on internships, yes, summer internships are extremely useful too. And I was able to participate in the Leadership Alliance, for instance, back in 2005. And that was a great experience. It's an internship based on, for me, in my case, it was biomedical research. But just to put it out there, before joining grad school, in my case, I performed molecular biology research. So not that much related to the current type of research that I do nowadays, but it's still science. It's still using a set of tools to answer a problem. So getting those skills honed in over time, doing different sets of sciences actually helped me a lot. So get those summer internships; those are really useful, and vary a little bit the set of skills that you're gonna develop in those.
Yeah, just like to piggyback on Willy and what Irena said earlier, is that try to expand your toolbox. Try to learn different skill sets. Don't always be funneled into one set. The more exposure you get, like Irena mentioned that she would go on the weekends as well. I wouldn't recommend a 2-hour drive, but I would say,
Neither would I.
The more exposure you get, and the more you could always reach out. One thing I always had difficulty was I never used to ask as many questions because I was always shy. Just feel free to ask the questions, and I know a lot of the mentors would love to answer the questions, and the more interest you show, it's more willingness them to teach you more and give you that responsibility to like expand your own knowledge base.
I would say as much hands-on learning as you can get is the best you can. As many labs as you can get are the best. All my lab-based classes were the best I could. It really helped you out. Book learning is really amazing, but until you can actually do it with your hands-on doing, it's a whole different ball game, because you can really read something in a book and know it, but until you actually do perform something, it's really a completely different situation, and you're not going to understand it until you're actually doing something.
Yeah, I think all of what my colleagues have said is definitely true. Try to get hands-on experience and try to explore different paths, different areas of science. So when I started in undergraduate, I was going in with the mind of going to med school. I was pre-med and driving all my energy into what do I need to get into med school. And it wasn't until junior year where I volunteered in one of my professor's labs, molecular biology, again, trying to just check off that, okay, I volunteer in a lab to try to get into med school. Then I realized that there's other options out there. You love science and you want to help people. There's other options of career paths besides med school. So I fell in love with research and just being in the lab and doing investigations and, learning every day. And, that's another way of helping people. All the work that you do in the lab eventually, goes on and it's out in the clinic and somebody's, taking the medicine that you worked on, and it's really fulfilling. So I would tell myself, just be open to other career paths, maybe earlier in undergraduate, studies.
Thank you. So I do see some questions coming into the chat. We will have a Q&A a little bit later on. We'll definitely answer those questions. From that, I got pay attention in chemistry class, do lots of internships, and get as much hands-on experience as possible. Can any of you guys share with us what you are currently working on at Regeneron in your particular department?
I am currently working on coronavirus every day, and then many, many other things. But yes, coronavirus is a constant.
Can you maybe share with us a little bit more specifically what that looks like in terms of how you're working on it?
I started working on coronavirus. It's been like a daily thing for me. I started working on it in January. So I was developing, I work in the immunoscreening group in therapeutic proteins. So I was developing assays to identify blocking antibodies. So I developed assays, from basically the base. So I developed the assays from the bottom up to identify the antibodies, and I do screening. So we have groups that immunize the animals and then do make hybridomas, and then I'm part of the group that screens the hybridomas that make the antibodies. So I'm part of identifying the antibodies, and then I do further analysis to identify the properties, and then they come back. So we have other groups that do further purification, and then I come back around with my assays to identify the properties of these antibodies when they're purified. So everyone works all together, but there's been a lot of things going on, and then there's some further later stage things that are going on, and also next generation, trying to identify some newer antibodies. So it's a constant thing, and lots of people are involved. But yes, it's been going on for a very long time, to say the least.
And I guess how often does that change, like your task, I guess, for the day? Does it change much?
Yeah, I would say a lot, depending on where you are, but tasks change, at least for me, day to day.
Thank you. I'll also open up another question to the group. Many of you mentioned what you love most about your job, but what do you like least about your jobs?
Stalking the lab. Sorry, Willie, I cut you off. Go for it.
No. No, yeah, I mean, what I don't, the least stuff that I like about my job is that you don't have sometimes enough time to do more. There's so many cool questions that you could ask in so very little amount of time that you can invest into answering that questions that forces you to prioritize the type of questions that you need to ask. And Pam is my supervisor, she can attest to this. I would come into her office and would come in with a million different questions. I would drag Irena with me along also, and we would brainstorm for 20, 30 minutes, and Pam would be nodding, and she would tell us like, how does this push the product forward? And this is a good question, and we could justify that, but in reality, it might not really push it forward. And sometimes you need more time, and often it's unrealistic sometimes, but in a perfect world, if time would be limitless, I guess, that would be great. But in the meantime, prioritization is a good skill to learn. So you can practice it from now. I'm pretty sure you're all taking 4, 5, 6 classes. And you got to prioritize what to learn and what not to worry about. And that comes really helpful a little bit later on. So not because it looks cool, it needs to be done right away. That's take home message from my part.
And I see Sangrim and Ishita smiling.
No, I agree with what Willy said. The changing priorities are important, and also we are very deadline driven. So it's like we are all working as a team. So what I'm doing, the next group is waiting for my results. So there is, and also we are not working on one project. In a company, you're working on multiple projects. So the time to do independent research is maybe a little limited, but as everyone is saying that, it's an extra time you have to devote if you want to do it. It's not like, we are very busy, but we all enjoy being with each other, enjoy each other's company. We do the hard work, we meet our deadline. We always do that. Yeah. We work hard and we play hard.
Yeah, there's definitely a lot of things in the Regeneron pipeline that you want to get your hands in and get your work on, but sometimes that's because of priorities, and like these guys mentioned, priorities change and you have to shift gears a lot. I think that's something you have to just adjust to and wait for the opportunity when you can work on these projects. But there's so many things in the pipeline that's so exciting, and eventually you'll get to work on them. It's a patience game. You just have to wait for it.
That's good. Well, I want to thank our panelists for sharing with us. I'm going to turn it over now back to Sarah.
Oh, Sarah, you're muted.
I can't hear you.
I can't hear you.
She's not muted. Oh no.
Nope.
Oh no. Well, did you want one of us to move along with your?
Sometimes you have to leave and then come back, and then the sound works again.
Technology.
Okay. Well, we'll get moving on. Panelist 2 that Sarah's doing are scientists creating therapeutic drugs in early stages. And again, we'll have everyone in that panel go around and introduce themselves. That's starting with Jacob.
Sure. Hi, I'm Jake. I've been at Regeneron for over 3 years, and I currently work in the neuroscience group. And I really like working at Regeneron because I get to learn new things every day, and I get to work on medicine that's going to help a lot of people, which is really exciting stuff.
Jen? Jen, do you want to go next? Oh, Jen, I think we just lost Jen. How about Scott? Oh, Jen, are you back? You're on mute.
Yes, yes. I think it's yeah. My internet has a problem, so I have to move to the other room. Yeah. Okay. So my name is Jing, and I have been working in the Regeneron for 16 years, over a little bit over 16 years. What I love Regeneron is not just because Regeneron leading the science and find ways to innovate and continuously improve, but also I love the environment. I love the we can be work together, and then as a teamwork and collaborations, and we always try to our colleague will not just work as a group, and we also have a lot of other groups try to help, not just for working. And then region like a melting pot. We have people from different countries and different backgrounds. We always like the culture is not just for one culture. We actually be able to accept about other people's. It's we have the race and the how to say that we have a different one. We in Regeneron, like you guys, we celebrate the Indian New Year. We also celebrate the Chinese New Year or the other like African Americans the good festival ceremony. So it's a very good place to working here. I love it. Okay, thank you.
Scott?
Yeah, hi everyone, Scott McDonald. I work in the cardiovascular research area. And my group actually runs sort of all the cell-based assays for the group. And this includes, assays exploring heart failure, fibrosis, which is a scarring disorder, kidney disease, and pulmonary disease, so different lung diseases. So we do a lot of things supporting a lot of different therapeutic areas and disease areas. And that's sort of, what I love about what I do. We have a wide range, a set of assay systems, models that we use in my lab, and to do, like the video said, basic biology all the way through to the clinic. So it's really exciting that we have the tools, the resources, and the freedom to do basic biology work, starting from human genetics to explore and identify new areas and ways to intervene in all these different really important areas of research and need, since there's so many patients with cardiovascular disease out there that need additional medications to help manage their course and improve the outcome. So it's a great place to work. I've been here about 5 years. I've worked at another company before, and I can say, the tools, the resources, everything that people have said about Regeneron is true. Believe the hype. We do it. We lead by science. We work as a group. We think outside the box. The technology is exciting and moving quicker than you can keep up. There's more to do than we can do, but it just keeps every day pretty exciting.
May?
Yeah. Hi. What I like most about Regeneron is it's really like this company is really science based, and then our products it's generated, it's in-house generated, not like the other big farmers. They tended to bought from the other small companies. So because I had previously I was transferred from the West Coast to the other big pharma companies. So I have truly feeling about the difference between Regeneron and the other big pharma companies. So this company truly is science-based, and also the culture for this company is really science-based. You can tell from the founder and the co-founder still the original. They started the company, so they are really like the company. They truly science-based. So that's what I like the most about this company.
Grace?
Hi, I'm Grace. I've worked at Regeneron for 2 years, and I'm part of the immunization group, where I work with mice in order to develop antibodies that hopefully become therapeutic treatments. And I really enjoy what I do because I have the opportunity to be part of developing a treatment that could potentially change people's lives.
Hey, Suk.
Hi, I'm Jesus, and I was also working with the same group what Grace just described. And I have been in Regeneron like around over 4 years, and then almost 5 years anniversary next year only. And as you saw the video, we have a very valuable mice available to generate a monoclonal antibody. And you heard about from the coronavirus antibody we generate. So actually the role at Regeneron, for my role in Regeneron, we are the first place actually, we're moving forward to generate antibody. So Scott talked about the cardiovascular, he developed some target and we can generate antibody, and then it came to us and we're using all the available source or tools to generate the best antibody. And then I'm doing as a first place to generate that antibody in here. And this is my first company, pharmaceutical company. Actually, I came from long time academia background to move myself to the industrial background. And then I feel like I'm very successful to move industrial background from academia. And then because Regeneron, as all our, the members talk about earlier, Regeneron is the best place, I think, to work. And sometimes science, to see outcome, it takes really, really long time. It looks like an endless marathon game. But sometimes it looks great. What Kristen just said, we were able to make antibody for coronavirus within a year. So sometimes science can be short, sometimes it's too long. So everybody just, hope during this, the virtual, meeting to hear about all science, how can be wrong, to be patient together, there's some come out, or how we can be lucky to have a good thing come out to use. I think industrial pharmaceutical industry is a really good place to see, how our hard work, how our long marathon can affect people's life. Some people can smile, some people can live really happily for their life. I really happy to see their face with a big smile. I can change their life, or we can change their life. That will be very great to do science in here. I'm done, Pam. It was wrong.
No, you're fine.
Thank you so much. Can you hear me now? Can you hear me now? Yes. Oh, okay, great. Thank you. So I would like to ask the volunteer to talk about their career path from panel 2, from college to where they are now. So Kim, I believe we have someone sign up ahead of time.
Yes, so Jake will be taking us through it.
Great, thank you, Jake.
No problem. So I graduated from Gettysburg College in Pennsylvania in 2013. And while I was there, I studied psychology, neuroscience, and music. While I was at school, I knew I was interested in science, but I wasn't really sure what area. It's similar to what we heard from Irena earlier. And when I got to school, I took a bunch of different, and this is kind of the beauty of going to a liberal arts school.